Hi everyone, thanks for being interested in my research. My name is Olivia Dickinson and I'm a senior at Providence College where I study applied physics and math. And today I'm going to be talking about the experimental research that I undertook at Duke this summer via the RU program at Tunnel. And so what I worked on was experimental planning for measuring the cross-sections of short-lived radionuclei. So basic nuclear reaction cross-section data such as N2N, N-gamma, and gamma-N producing short-lived isotopes with half-lives between seconds and minutes are critically important to a breadth of scientific fields, including applications relevant to national security, medical isotopes, fission and fusion reactor data, etc. However, not much experimental data is available due to various associated limitations, including poor counting statistics. The limitation can be overcome by using the cyclic activation technique by way of a fast irradiated transfer system. With the availability of a fast transfer system, namely rabbits at Tunnel, which I will talk about later, the cyclic activation technique can be used to study such reaction cross-sections for nuclei having half-lives between seconds and minutes. The primary isotopes of interest in my experiments were germanium-73, germanium-77, molybdenum-91, indium-114, and indium-116, and will be produced using quasi-monoenergetic neutron beams from the 10 MeV tandem laboratory at Tunnel, where we house the Van de Graaff accelerator. An array of highly efficient and broad energy germanium detectors will be used for the gamma ray counting, and a detailed energy and efficiency calibration of the detector array, along with the conceptual and computation work related to the project, will be presented in my PowerPoint presentation. So like I mentioned, we're able to undertake this research measuring the cross-sections of short-lived radionuclei at Tunnel because we have the rapid belt-driven irradiated target transfer system, which we call rabbits, and this allows the possibility to transfer short-lived isotope samples to a counting room with limited background radiation. Previously from this, we would take the irradiated sample, having a half-life of a minute or longer, and run it to a different room where we would count the gamma ray emission from the irradiated sample. But this allows us to look at samples that have much shorter half-lives. And so there's three ways that we can produce neutron beams in the tandem lab for energies that we would like to use for our experiment. The first way is going to produce a neutron beam with an energy of 1 to 4 MeV. So we take a proton plus a tritium atom and collide them. And the collision produces a neutron beam with energy between 1 and 4 MeV and also a helium-3 atom. A second way we can produce a neutron beam, and these neutron beams will have energy between 4 to 13 MeV, is by taking a deuteron and a deuterium atom, colliding them, producing that neutron beam with an energy of 4 to 13 MeV plus a helium-3 atom. Third way is going to produce neutron beams with energies greater than 14.8 MeV, and that's by taking a deuteron plus a tritium atom, combining them, producing that neutron beam with an energy, like I said, greater than 14.8 MeV, and then a helium-4 atom. So those quasi-monoenergetic neutron beams are what we use to begin the radiation of the sample. And so the Van de Graaff accelerator is located in the tandem lab. And so there's a neutron beam line that heads straight to the neutron time of flight room where the samples are radiated. In that time of flight room is where we see the rabbit system. The rabbit system brings that irradiated sample through the wall to the counting room at a very high speed in order to count it immediately. And so my research is just focused on what isotopes do we want to look at, which I mentioned are those 4 before. Then I'll be talking about how we calibrate the gamma ray detectors and gamma ray spectroscopy in general, and a little bit more into the rabbit system and why those are the 4 isotopes of interest. So thank you. I look forward to answering questions about my research.